Okay, now let's talk about the rest of chapter 14, sections 4 and 5. So, how do parasites affect larger ecological communities, well, populations and communities for that matter? So, indeed, a parasite or pathogen can have a significant effect on a population, causing that population to decrease over time, as is shown with these ladybugs, ones that are unaffected and they have relatively high fertility rates, and ones that are affected by a parasite and have much lower fertility rates because of that. And in some cases, a pathogen can essentially drive a species to extinction, which is what happened to the American chestnut. Now, the American chestnut isn't actually extinct, but from an ecological perspective, it's really extinct because it was very much a dominant species that ranged over a good chunk of the country, but now is virtually gone from wild populations because of a fungus that was introduced from Asia. You can see from this old picture, they used to be very large, dominant trees. They were very important for wildlife because of the nuts they produced. And so, because of their absence, other trees have had to take up that space. And so now the forest community is quite a bit different than it was at that time. Um, all right, so population dynamics can be influenced. Here's a situation. This is a species called the grouse, the red grouse. And up here in the upper left, this shows two populations that exhibit what is a normal population cycle for this species, in which they crash about every 4 to 5 years. So researchers hypothesized this was due to a parasite that they had. And so what they did was they essentially treated some of the populations um, with a drug that would kill this parasite. And so here are two populations that were treated in 1 year, 1989, and here are two populations that were treated in 2 different years. And I don't know, what do you think? The conclusions of the authors is that reducing the parasite or removing the parasite from this bird essentially reduces fluctuation, which is somewhat easier to see in this graph than this one, I think, in that you can see there's not quite as much up and down, although in 93 it still went down quite a bit. So their conclusion again was that the parasite influenced this population cycle. Now, parasites can also influence the relationship between different hosts, and so this is the flower beetle, Tribolium castaneum and Tribolium confusum. So these are two competing species that feed on flour and other grains and such. And so you can see when there's no parasites in either population that castaneum is clearly the dominant species, whereas when both types have the parasite, confusum is the dominant of the two, um, changing the competitive outcome of these two species. Apparently, confusum can better handle the parasite. When the parasite's not around, castaneum is simply a better competitor. Having parasite load can influence a species in different ways. So, for example, with the moose here, they have to have very high fitness in order to withstand the predation of wolves. And so individuals with a higher parasite load, lots of ticks, moose are apparently notorious for getting lots of ticks, can compromise the overall health status of that moose and make it more susceptible to being preyed upon by the wolves. And we talked earlier about how this parasite Saculina has a negative impact on crabs and really changes their behavior, such that now instead of the female producing eggs, she just produces a bunch of the juvenile Saculina, but yet she behaves in the same way as if she were producing and releasing her own eggs, and also to the extreme of causing the males that are infected to basically act like females. The physical environment that the animals live in can be affected. So here's this crustacean, this small shrimp-like creature that lives in the mud flats, the sediments of marshes, estuaries, coastal wetlands, essentially. And when the parasite of Corophium, this crustacean, is not present, what happens is the Corophium live in these clusters that stabilize the sediment and cause it to build up in these tiny islands. But when they are infected with a parasite, the Corophium population goes down quite a bit. And what happens is these islands then become less stable and there's greater erosion. Erosion increases and the silt content of the mudflats decreases. So this is an example again of where a parasite has a negative effect on a population, which then has an effect on the physical structure of the environment. All right, let's look at section 5. So here we're going to talk a bit about how we do some mathematical modeling of host pathogen dynamics. And so here are our variables: number of susceptible individuals, number of infected individuals, the transmission coefficient, that is how easily is it transmitted from one individual to the next, death and recovery rate, essentially the rate at which infected individuals are no longer infected. They either leave the population through dying, or they basically recover and then are immune from the pathogen. Susceptible individuals have not been exposed to the pathogen yet. S and I together are basically the probability that these two types of individuals are going to interact with each other, and then when you throw in beta, that gets at not only how likely are they to interact, but then also give the disease to each other. And when we have the spread from one individual to another, we generally refer to that as horizontal transmission, but you can, with some cases, speak of vertical transmission, which is the spread from a mother to a child. For example, HIV, the human immunodeficiency virus, can be spread by a mother to her baby during childbirth. Some diseases, some pathogens have a latent period, as we've seen in the news with Ebola, that you can have in that case, the virus, but not be spreading it yet. That's, again, the latent period, as we've seen in the news, which is anywhere from 2 to 21 days for Ebola. All right, so let's look at our equation here. So this is essentially the change in the infection, number of infected individuals in a given unit of time, is a function of how many infected individuals there are, how many susceptible individuals are, and that transmission coefficient. So this side adds to the spread of the infection, the number of infected individuals. And this side is, of course, that recovery rate times the number of infected individuals. So that will decrease or take away from the number of infected individuals in the population. So, and looking at that equation, of course, if the left side is greater than the right side, or if overall this equation is greater than 0, then you'll get the spread of the disease. If it's the other way around, you'll get the decline of the disease. Also, you'll get the spread of the disease when the number of susceptible individuals is greater than essentially the ratio of the recovery rate to the transmission rate. And this is sometimes referred to as the threshold density of the population. All right, so essentially to try to control a disease, you want to control or reduce the number of susceptible individuals, because then you can decrease the chances that infected and susceptible individuals are going to get together. All right, one very crude way of doing this, of course, is to literally remove or eliminate susceptible individuals. And this is done with livestock. If you have an outbreak of chicken or cattle in a particular herd, to keep it from spreading to other herds, you just simply eliminate that herd. You cull the herd, slaughter those individuals. Now, in people, we will vaccinate individuals because, particularly for viruses, we do this. So when someone is vaccinated, they are no longer in that susceptible group of individuals. And that can have a impact on the spread of the disease. So in this example below, this is for measles, this communicable disease, and these are data particularly from Romania. And so they started an immunization program in the late 70s, early 80s, and you can see that the percentage of people being vaccinated increased rapidly and reached a pretty high rate. Before the vaccination program, the number of cases was quite high, but afterwards it was quite low, although you can see it still fluctuates. They have some years where they get particular outbreaks, but nothing like the pre-vaccination days. You can also address the recovery rate, and obviously individuals who get infected, you want to get them uninfected, recovered as quickly as possible so that they are no longer have the ability to spread the disease. You can quarantine individuals. Essentially, this is going to decrease the transmission rate, so by isolating those infected individuals, you'll keep them from spreading it to those susceptible individuals. Okay. Now, what things can influence the spread of disease? Well, it's thought that a changing climate, or as the temperature increases, that can promote the spread of certain pathogens from more southern regions to more northern regions. As the climate basically shifts north, or it gets warmer further north, those diseases that were sort of restricted to the tropics beforehand can spread. And this disease in particular, it's spread by these type of rodents that live further south, but they could potentially spread further north with a warming of the climate. Beginning of the chapter talked about the enslaver parasites, the parasites like Saculina that change behavior. Here's a couple more examples. This is a parasitoid wasp that parasitizes spiders, and so it lays its eggs on the spider. And in this case, not only does the larval form of the wasp feed on the spider, but it changes the spider's behavior. On the left, we have a normal spider web, but on the right, we have one that is from an infected spider. And so you can see that the web is much more different. And the spider will cocoon itself in a mass of spider silk, and then the larval wasp will essentially feed on that entombed spider, if you will, eventually breaking out. So instead of building a web to catch insects to feed on, it basically builds a web that brings about its own demise, you might say. And like that first example we saw at the beginning of the chapter, here's a pathogen that in these this insect called a gypsy moth. These are the caterpillars of gypsy moth. The pathogen that is parasitizing, that is living in this host species, the moth, has a particular gene that modifies its behavior. So what we see here in these data are essentially 6 different experimental groups, and the WT represents the wild type or normal form of the virus, and it modifies the behavior of the moths to climb higher, which facilitates spread of the pathogen. They hypothesized that there was this particular gene, abbreviated EGT, that is responsible for this behavior. So what they did was they engineered some of the virus that did not have this particular gene, and some of the virus where they removed the gene and then reinserted it. And you can see the one where the gene has been removed, those moths do not climb nearly as high, which would make the pathogen less likely to spread, a virus in this case. And those that were re-engineered to have the gene act like the wild type virus, the normal type virus, promoting higher climbing and facilitating spread of the pathogen. Okay, that's it for chapter 14.